Good morning, guys. Welcome to my channel. So today we'll be talking about cellular aging. So first, let us talk about the definition of cellular aging. So Robbins says that it is basically a result of progressive decline in cellular function, viability caused by genetic abnormalities and the accumulation of cellular and molecular damage due to the effects of exposure to exogenous influences. You can clearly see that basically in cellular aging we are focusing on the damage that is caused to the cell. Okay, what are the damages that can be caused? Cellular functions and viability can be disturbed, and these can be caused by genetic abnormalities and accumulation of the cellular and molecular damage due to exposure to certain influences, which can be exogenous influences, right? So what could be the exogenous influences? Now let us see about the potential causes for the cell aging. First one is the DNA damage. Secondly, we have cellular senescence. Third, we have defective protein homeostasis, and fourth, we have dysregulated nutrient sensing. So these are the four important side endings you need to remember, right? Right. Now let us talk in detail about DNA damage. So what happens in DNA damage? See, Robbins has given a statement like most common hematologic malignancies are diseases of the aged. So what do we mean by this is actually what happens is normally in a cell, okay, if you take a normal cell, it undergoes senescence, right? It undergoes several stages until it reaches the senescent stage. That's the last stage. So until it reaches that particular stage, that's the hematopoietic stem cells will suffer many mutations, right? So if you take a normal adult cell, it will undergo many mutations, and Robin says that they undergo 14 new mutations every year. So because of these accumulation of the mutations, what will happen? There will be certain DNA damage that occurs, and this DNA damage will be accumulated over the time. Right, that is the reason he says that most of the hematological malignancies are diseases of the aged. Like most of the people who are aged, they will be having these hematologic malignancies. Right. Now let us talk about a syndrome called Werner syndrome. See, Werner syndrome basically means that it occurs in people. Okay, like it occurs in most of the people, and they who are undergoing premature aging. So it basically it means that a child is undergoing aging in an early period in his life. So that's referred to as the premature aging. So the defective gene over here is DNA helicase. So basically we know DNA helicase is involved in DNA replication, DNA repair, and DNA unwinding. So whenever there is a defectiveness in this DNA helicase, that lead to the syndrome called Werner syndrome. Now he has also explained about two important other syndromes called Bloom syndrome and ataxia telangiectasia. Right, these are all about DNA damage. Now let us talk about cellular senescence. So cellular senescence basically involves two important characteristics. First one is called telomere attrition, and the second was called activation of the tumor suppressor gene. Now, what do we mean by telomere attrition? Before we come to this particular topic, let us understand what do we mean by telomeres. So basically, telomeres are short repeated sequences of DNA that are present at the end of linear chromosomes, right? And there is an enzyme called telomerase, which is responsible for maintaining the length of this particular telomere. So, but basically, telomerase is a specialized RNA protein complex. It uses its own RNA as a template for adding the nucleotides to the end of the chromosome, right? So, this telomerase is expressed in germ cells and is present in lower levels in stem cells, but is absent in somatic cells, right? Now, we'll talk about the mechanisms. If you see this particular picture, you will understand that this yellow color portions refer to the telomeres. Okay, now if you see this end, actually I have magnified this particular image here. You can see that there are certain repeats called TTA triple G repeats. So these are basically the telomeric repeats which are present at the end of the chromosome. I already told that this telomerase enzyme is responsible for maintaining this length of the telomere. Now, as we age, right, when the ages increase, and there will be repeated cell division, and there will be telomeric attrition. This occurs normally in all the cells, like in all the human cells, including you and me. So what will happen? The telomere length will shorten. Okay, you can see over here, the yellow color has decreased in length. Now what happens? There will be defective cell. Now the cell is considered to be defective. It proliferates, and the DNA damage response will be there, and because of which there will be senescence of the mature cell and depletion of the progenitor cells. This actually occurs in normal humans, right? This part occurs in all the humans. But if you take in the case of cancer cells, what will happen is there will be something called as the telomerase reactivation. Actually, telomerase will help in maintaining the length of the telomere. Now in these normal cells, what will happen? The telomerase activity will not be there. So what will happen? the telomere length will decrease in size. But in case of cancer cells, the telomere's activity will be increased, and because of which there will be increased the length of the telomere will be maintained, and because of which there will be senescence of the mature cell will be absent. So cancer cell will proliferate. So that is the most important characteristic of cancer, right? Metastasis. Right. Now let us talk about activation of tumor suppressor gene. So if you see that there's a gene called CDKN2A. So this basically is the tumor suppressor gene. It encodes two proteins, P16INK4A and P16ARF. These both are tumor suppressor proteins. So basically they prevent uncontrolled mitosis and they push the cell towards senescence. Right. Now these all are necessary for the humans, right? Because we need not have uncontrolled mitosis, and we should not make the cell to live for long, right? We should make the cell to die at one particular time. Otherwise there will be increased number of cells in the body, which will lead to cancerous conditions. Now let us talk about the third point, that is the defective protein homeostasis. See, if you see here, there are two important things you should remember. First one is maintenance of the protein correctly folded configuration. See, when the protein is folded in correct configuration, we can maintain the protein homeostasis basically. So this is done by a component called chaperones. They basically help in maintaining the configuration of the proteins. And we also need to degrade the misfolded, damaged or unneeded proteins. Okay, how is it done? It is done by two mechanisms. One is the autophagy, or that is by the lysosomes, and the other is by the ubiquitin proteasome pathway. Right. If you see over here, he has also mentioned a drug called rapamycin. What does this rapamycin basically do is, it inhibits mTOR. mTOR refers to mechanistic target of rapamycin. So rapamycin will inhibit mechanistic target of rapamycin, and which in turn slows down autophagy. So if you see clearly through this pathway, you can understand that rapamycin will actually cause atrophy of the damaged proteins, right? Because rapamycin inhibits mTOR. mTOR will in turn slow down the autophagy. So we can see that rapamycin basically causes autophagy of the damaged proteins. So basically cells will live longer because of this, right? So this comes under defective protein homeostasis. Now let us talk about dysregulated nutrient sensing. So that is basically a hypothesis, you can say that, like most of them believe that, or even scientifically we can believe that, but not much proof is present. Like people who eat more, right, throughout their lives, they are more comparatively to get aged faster compared to the person who eats less, right? So that comes under dysregulated nutrient sensing. So basically the insulin is there, no insulin or IGF-1, we need to decrease that, and serotonins need to be increased. So these are the main concepts behind dysregulated nutrient sensing. If you talk about first case, IGF-1 signaling pathway, they are produced in response to growth hormone. That's basically by the pituitary gland, growth hormones are produced. Because of their response, this IGF-1 is produced. IGF-1 refers to insulin-like growth factors, right? Now, if you lower the IGF activity, what will happen is there will be increased lifespan, reduced cell damage, and slow metabolism. Now, IGF-1 is basically a target for mTOR. I have already discussed about mTOR here, right? mTOR I have discussed over here. So basically IGF is a target for a protein called mTOR, which will in turn slow down autophagy, thereby increasing the cell life. So now you already know that mTOR is blocked by rapamycin, and hence lifespan is increased. Now let us talk about the second thing called serotonins. We need to increase serotonins, right? In mammals, basically he has given that there are 7 types of serotonins are there, and he has also described about serotonin 6. We'll see that in further time. Right, so there are 7 different types of serotonins in different cellular compartments. And basically what are serotonins? They are proteins that help the cell to adapt to various environmental stress, food deprivation and DNA damage. So basically they turn their genes on and protect the cells and slows down the metabolism. They turn down harmful processes like apoptosis and damage from free radicals. They increase the glucose sensitivity and the glucose metabolism. Right, so they are basically helping in the cell, they basically help the cell not to die. So cellular senescence can be prevented. And serotonin, if you see, they enable caloric restriction, thereby it's a metabolic adaptation, and they also maintain the genomic integrity, and thereby by activating the DNA repair mechanism. So these are the 2 important functions of serotonin 6 particularly, and basically serotonins help in delaying senescence and the cell won't die so soon, right? Because it helps in the preventing the cell from, or basically helps in adapting the cell to various stresses like DNA damage, food deprivation, and environmental stress. Right, that's all about cellular aging. Thank you for watching.